So basically that today I think I'm going to more present to what we know that we develop better, and for those new project, I think I hope that I can report in the future. So my lab has been primarily work on the ischemic heart disease, and as you know that after ischemic injury, each time such as in a myocardial infarction, there are a lot of bone marrow derived cells flux into the injury, and especially immediately after the myocardial infarction, a lot of this kind of inflammatory cells are maybe progenitor cells that these cells actually very important impact on the long-term outcome of this very devastating disease. And I think we are very interested in the bone marrow progenitor cells because these cells are very, very important. For example, from this JCI paper published over 10 years ago, showing that if you have a deficiency in the c-KIT, it's a stem cell growth factor receptor. If you have a deficiency in the c-KIT in the bone marrow cells, then you make a myocardial infarction in the heart. The heart is doing very, very badly and cannot recover. And so, at the same time of this basic research evidence, there are also a lot of clinical observations. Like over this 10 years period of time, quite a few publications in the New England Journal of Medicine showing in patients with ischemic heart disease, if you have a lower circulating bone marrow-derived progenitor cells, you can very reliably predict a very bad outcome and increase the mortality. So we believe these progenitor cells of bone marrow derived, they are very important for the outcome and for the tissue repair. So perhaps the mechanism is very complex, but the importance is very well established. And in a way, I think we are actually, in the basic science, we kind of a little bit study in this aspect. So my lab, one of the projects is primarily focused on the contribution of bone marrow cells to the ischemic cardiac injury. And we believe that after myocardial infarction, ischemic heart disease, and a lot of physiological factors that are released actually act on the bone marrow microenvironment to release the cells, including the progenitor cells, out of the bone marrow and circulating into the blood and coming to the ischemic tissue for the repair or for the damage. So this is in the bone marrow microenvironment. My lab has worked very extensively that how these stem cells are returned in the bone marrow, how an ischemic condition that they are mobilized, these stem cells that no longer be able to return in the bone marrow get released. And we are able to establish that even though there are so many interactions between the stem cells and the microenvironment, the niche environment, actually we were able to establish that these three interactions between these two components are very, very important in the adult life. And such as this pair of integrin interaction, this pair of chemokine and the chemokine receptor access, and also this c-KIT and stem cell factor and its receptor, this receptor tyrosine kinase, I think we believe these are the most important, and each of them actually has a lot of development pharmacologically in order to mobilize stem cells, and for different purposes, not only in the cardiovascular system and also in the hemopoietic system. For example, that especially in this one, that the 6L4 antagonist has been used in the clinic now to mobilize stem cells for bone marrow transplantation, especially for those patients that are resistant to stem cell mobilization by GCSF. So this is important for stem cell mobilization. So we were able to establish the crosstalk between these signaling pathways. These are so important that to maintain the cells in the bone marrow. Yeah. So our lab has also focused on that when the stem cells are recruited into the ischemic heart, how they receive the signaling for migration, for adhere, and for invade the ischemic tissue, and how the progenitor cells be able to differentiate into vascular cells, for example. And we believe these stem cells are important in a way that, especially for the vascular tissue, because of the similar and very late branch of the hemopoietic system and the vascular system, so they share the similar stem cells developmental traits. And also that they can invade into the ischemic tissue to exert their effect in a way that normal angiogenesis, the vascular branching type growth, cannot compensate. So, and one of the aspects that we realize that progenitor cells differentiation is actually very important. As you all know, that there are certain stem cell therapy trials for the ischemic heart disease with different cells, mainly bone marrow-derived progenitor cells, and also a few cardiac stem cells. And we are primarily interested in the bone marrow progenitor cells. And these cells that there's a discrepancy in the clinical observation and also their experimental evidence. That is that you always see a certain level of benefit, but you do not always see that these cells really physically become the vasculature, because that people think that the main effect of these cells are paracrine. So they may secrete some growth factors, some survival factors that have an effect, even though the effects are modest in the clinic. And so we are interested because of that. So if their differentiation into vascular cells is very modest, is very limited, so I think this may be a barrier for the stem cells to have a full functional effect. So in that case, that this discovery was made recently, as already published last year. So I just go over very quickly, and we come to this discovery because of E12 family transcription factor. So this family, as you all are aware, this is very important in that cell cycle checkpoint control. Basically, RB-E12 family. Now, our lab has worked extensively on these transcription factors. They are unconventional beyond the cell cycle function, especially the cell type specific role in the vasculature. And for example, E12 is supposed to be a cell cycle promoter. They are actually very important for the gene expression that are important for the DNA synthesis for cell cycle. And, but however, even though this is a positive regulator, but in the knockout we have been working on that, and we established that actually you knock out the E12, you have an enhanced angiogenesis. So it's kind of unconventional as its role in the tumor. And we also shown that the E12, you knock out that it's actually have a increased blood pressure. Then we find that only E23 seems to be absolutely required for the cell cycle in the vasculature. So you gotta knock out E2/3, the vessel are no longer be able to grow. So basically now we establish that. And so since we are stem cell people, so we come more a little bit focus on what it does, E2F1 for the bone marrow stem cells. They have a knockout, have a better angiogenesis. So we did perform microarray for the at that time for the E2F1 knockout cells compared to the wild type. We find that actually out of this cell cycle gene knockout, we didn't find any difference in the cell cycle gene expression. So we find that over 75 to 80% of the genes that are altered actually are metabolic genes. And so we were not able to dissect upstream, downstream. But one fact is very obvious, is that this is PDK, that this pyruvate dehydrogenase kinase 2 and 4 are very low. And this has been confirmed by Western, by RT-PCR, etc. So I mean, all know better than me in this oxidative metabolism that you have a reduced PDK, you would have a released inhibition and the pyruvate dehydrogenase. So you would have an increased acetyl-CoA, have increased mitochondrial oxidation. So actually, it is the case that when we perform this seahorse analysis, we find that these knockout mice stem cells do have an increased oxygen consumption and actually seems to have a reduced anaerobic glycolysis. So this is very interesting. These stem cells have such a, so I think the paradigm is that normally we believe that when the stem cells primarily rely on glycolysis, when they differentiate, they actually adapt to oxidation, more and more oxidation, become functionally mature. So there's more mitochondrial function. And so this model seems to give us some suggestion, okay, what if otherwise? Whether actually you change the metabolism by knockout this gene, basically make them all more like oxidative. So what's wrong with whether there's change in the differentiation? Okay, so this might not be, maybe we double way. Maybe the oxidation adaptation is not adaptive. Maybe actually also itself actually have a role in the differentiation. So we want to test this hypothesis. We find that indeed, actually in this knockout mice with a high oxidation, actually these cells we culture in the endosenial differentiation medium, these progenitor cells are easier to become endosenial cells, and as shown by this marker. So these cells actually easier to become differentiated into endosenial cells. And actually that if we transplant these knockout cells marked by this GFP, so we transplant these GFP and the E2F1 knockout bone marrow cells into the wild type mice without any genetic marking. So then we make a myocardial function in the heart. So all the green cells are from bone marrow, all the red cells are the vessel. So we find that the mice that we receive this type of bone marrow have a much more bone marrow derived cells that incorporated into the vasculature, suggesting that these cells are actually more prone to differentiate into vasculature. So this is very exciting. I think the effect is kind of dramatic in our view. So actually, then we perform a lot of bigger sample size that replace the wild type bone marrow with the knockout. And actually, we were able to show that if you change the bone marrow totally with the E2F knockout cells, actually we can get the ischemic heart this mice with myocardial function can function recovered much better in terms of this fraction shortening and ejection fraction is contractile functions, and also that the infarct size is reduced, and the bone marrow derived vasculature is increased, and the total vasculature is increased. So I think that this part of the talk, I think that give us a lot of things to think about it. Maybe in this why this bone marrow stem cell therapy trial may not work, maybe because these stem cells actually are not able to differentiate. This everybody know. But we think that why they are not able to differentiate into the vasculature is because that they still rely on the glycolysis as a metabolic mode when they are already recruited in the ischemic heart. So they are lazy. They actually stay in that state. So maybe if we want, we can in certain way to artificially reprogram to manipulate their metabolic states before we put it into patients. Maybe we could enhance the effectiveness of this therapeutic strategy. Yeah. So then I think the other major part of my talk, I'm going to introduce as my new finding we made in the last couple of years or so. That is about, we talk about the bone marrow and the ischemic tissue. Actually, we know a lot of factors that are released from the ischemic tissue serve to a messenger mediated crosstalk between these two tissues. But recently that the fellows in my lab, they are actually very interested in the other particular molecular or particular mediators, and particularly the microRNAs and the exosomes. As we all know, this kind of become very important emerging field, and the reason we are thinking about that is because this microRNA, there are a lot of microRNA actually expressed only in the cardiomyocyte. We call this myocyte-specific mechanism myomere. Okay, these are not a trivial amount, expressed in a very high abundance in the cardiomyocyte. And when it happened, the myocardial infarction, they released in the blood, in the circulation, in the plasma, in a very, very high abundance. So clinically, that now this myomere actually are used as a biomarker of myocardial function, and especially these four, and 208A, 4995, 133A, and MIR1A. So very some, and so this is a lot of clinical observations. Overwhelmingly, these four actually are increase in the blood after myocardial infarction in patient. And the exosome is important because that now they increasingly realize that they are these small particles generated through endosomal biogenesis pathway, and actually about 100 nanometer diameter, 40, 100, 150, and also that one fact we observe is that they are very high abundance, normally or after injury. So they are very high level, actually, way much more than what we thought. So the first experiment that we did, we want to check the MIR after myocardial infarction. So basically, we induced the myocardial infarction in mice. We harvested the plasma. We analyzed this MIR. So as you can tell, compared to the sham and acute myocardial infarction, this is about 6 hours after myocardial infarction. So this microRNA actually increased exponentially, many fold. So we believe this seems not only the release, it actually have a positive synthesis process going on. And then we are very careful. because that you use U6 other RNA as for normalization when you analyze the MIR. So, but in the blood, we do not have standard nowadays. So basically, we perform some sequence analysis. We were able to pin down about 12 of microRNA in the blood that are always expressed consistently. So we normalize our increase of MIR by this consistent level of microRNA for normalization. So as you can tell that, then we analyze that whether this microRNA transport into a different organ. So we analyze different organ, at least for these 4 microRNA, we find that they are predominantly found in the bone marrow cells. This is 4 microRNA, and this is bone marrow. These are the sham. So after acute myocardial infarction, that this microRNA increased a lot in the first 12 hours in the bone marrow cell, mononuclear cells. That or some in the kidney, but not much in the liver or other organs, and also a manifold increase. So it looks they find a way into these bone marrow derived cells. And no, this is not because that after myocardial infarction, the bone marrow mononuclear cells themselves upregulate this microRNA normally expressed in the heart. Because that if we put this bone marrow mononuclear cells in the culture and mimic the ischemic hypoxic condition, actually this there's no difference. They do not increase. This is positive control in the heart tissue. Okay, so there's no difference in this bone marrow cells. They are not subject to the ischemic hypoxic upregulation in the bone marrow cells themselves. And also we made observation that the time course observation of bone marrow cells that contain this microRNA. And we find after myocardial infarction, the 6 hours, 12 hours, 24, 72. So we find that in the first 3 days or so, bone marrow cells always have this high level of microRNAs. So the second, we analyze the role of exosome, and then we have been very careful characterizing these exosomes by electron microscopy and size analysis and biochemical markers of these exosomes, make sure they are what we think they are. And that we find that primarily that these microRNA are carried in the exosome, and one of them seems more in the exosome depleted plasma, and the majority of them seems to be in the exosome. So basically, we analyze microRNA in the exosome versus non-exosome component in the plasma. So basically, there are a lot in the exosomes, and actually, when we culture, we did the experiment to analyze the transfer. So we isolated the exosome and the plasma depleted exosome, co-culture with the bone marrow mononuclear cells. And then we wash out and only analyze bone marrow cells whether this microRNA transfer. So we find that only your co-culture with exosome, not with exosome depleted component, that this microRNA get transferred into the bone marrow mononuclear cells. So it's clearly that seems exosome is way much more efficient in transferring this microRNA into the bone marrow cells in this cell culture system. And we also perform this in vivo. We isolated the exosomes from the mice after acute myocardial infarction versus mice from sham operation, and then inject intravenously into the recipient mice who have undergone nothing, so just the intact mice. So we find that only by doing this, by injecting the exosome from this acute myocardial infarction mice So we got the intact mice bone marrow mononuclear cells actually contain this kind of microRNAs. So that suggests that this actually be able to transfer in vivo. So we mark the exosome by certain chemical dye or membrane dye injected into the mice. And then analyze the bone marrow mononuclear cells by this FAHA analysis. We find that actually a lot of bone marrow cells are almost over 90, about 90 of the cells actually bone marrow mononuclear cells get taken up this labeled exosome. So the labeling, the uptake process seems to be very efficient. And actually we analyze the patient, how the patient. So we work with the cardiology, and we compare the patient with acute ST elevated myocardial infarction and the control patient, but without acute coronary syndrome. So we got two population of patients, isolate the blood exosomes, compare this microRNA level. So in the exosome of the acute myocardial infarction patient, their exosome contain way much more microRNA from the heart too. So this seems to be happening in the patient too. Then what's the molecular target? This is interesting because now this microRNA normally they have a defined target in the cardiomyocyte. Right? They are important for the development. Some labs seem to propose that a certain combination of this myocardial-specific microRNA can be used actually for some certain regeneration, protection. And now we are proposing that this microRNA get transferred into the bone marrow. Normally, they do not see this microRNA, whether there are some new target in these particular cells. So this become very interesting. If this is true, actually, we have a lot of exosomes, a lot of tissue-specific microRNAs. So this would be can be generalized that is another mode of exosome-mediated cross-tissue crosstalk. So it can be a common theme, a general theme. So we thought it's very important to figure out the bone marrow mononuclear cell specific target. So we did a lot of bioinformatics analysis. We were able to identify that different software platform. So we were able to identify that a lot of potential target, but one of them is particularly interest to us is that it looks like in the CXCR4 I mentioned at the beginning of my talk, we find that actually in the 3 UTR of this particular gene, we find that they have a site for the MIR1. So basically now we confirm that by mutagenesis of this 3 UTR reporter, so we were able to show that, transfect of this bone marrow mononuclear cells with this microRNA1, we were able to downregulate this report activity, and actually, if we mutate in this UTR, actually this MIR1 is no longer to regulate. It seems it regulates gene through this particular binding site, and we find that other 3 cardiac-specific microRNA do not seem to regulate this CXCR4 3 UTR reporter. So, but very surprisingly, if we actually overexpress either of these 4 cardiac microRNA, we were able to get it down of CXCR4. Okay, we were suspicious, so we use another cell type, the bone marrow mesenchymal stem cells. So we were able to observe the same phenomenon. So we are convinced that these 4, at least these 4 of the cardiomyocyte specific microRNA downregulate CXCR4, even though might be through different mechanism. And then this, I think, is another additional So whether this is, this I think is a protein level analysis showing the same thing. And not all the muscle specific, for example, this skeletal muscle specific, and these two, very close to what we just talked about, do not actually downregulate. So there are certain specificities there. And so we analyze whether this is exosome can do that. So basically we isolate the exosome from the acute myocardial infarction mice, co-culture with the bone marrow mononuclear cells that this is a sham exosome, this acute myocardial infarction exosome actually be able to downregulate the CXCR4 expression. And also if we treated the exosome donor mice with this microRNA antagonist, basically we inhibit this 4 microRNA altogether from the donor mice. Then we isolate the exosome, acute myocardial infarction exosome, put it together with the bone marrow mononuclear cells. Actually, this is kind of partially diminished. So at least I think that in the normal situation, acute myocardial infarction exosome be able to downregulate CXCR4 expression in the bone marrow cells seems to be mediated by this cardiomyocyte-specific microRNAs. And then we analyze this in vivo as well with a similar strategy. So we isolate from the mice with exosome from the myocardial infarction mice and actually inject intravenously to the recipient without any treatment. We were able to see that the bone marrow CXCR4 expression is reduced. And most importantly, I want to say what's the functional impact. So basically, here we would like to say the progenitor cells mobilization. So then we isolate that the exosome from acute myocardial infarction, just inject into the intact mice. And then we analyze that the progenitor cells in the circulation. So we were able to see that this is a lineage negative progenitor cell population. We were able to find that if the mice receive this acute myocardial infarction exosome, we find this recipient have a higher peripheral blood lineage negative progenitor cells. And this is diminished if you pre-treat the donor mice with the antagonist of this microRNA. And if we look at the secret cells, even more. And then if you now focus on the secret and the lineage negative, these very focused stem cells, very kind of purified stem cells, also are increased. So clearly that the treatment will be able to increase this stem cell population, presumably most likely from bone marrow. And also, since that the biomarker may not be very accurate, so we perform the colony forming assay that these stem cells be able to form the colonies if we put it in the limiting dilution, put it in the semi-solid culture system. Actually, by counting the colonies, we were able to also prove that the intact mice receive myocardial infarction exosome. They have a much more colony-forming progenitor cells in the blood. So basically, that proves this point. So I guess that this part of the talk, I show you that this exosome is really we are now more and more. I mean, another independent study. We isolate the human exosome from the diabetic patient, and then versus the normal individual. We injected in mice. We actually be able to induce a diabetic type of phenotype in the mice, a kind of glucose and insulin problem. So basically, that we are pretty much convinced that this exosome is a very big entity in the blood. Here I am not able to show. I use that as a cardiomyocyte microRNA to indicate the specificity of cardiomyocyte inference for the bone marrow cells. currently that we are in the lab that we are doing some genetic engineering method. We are using that the tRNA synthetase mutation. So after tRNA synthetase mutation, we were able to actually incorporate the amino acid during the protein synthesis, particular non-canonical amino acid in this engineering the cells. So because the mutation can be made in a tissue-specific way, so we were able to incorporate this artificial amino acid in the specific cells. And then we were able to use, because this artificial amino acid can be tracked, can be tagged by clinical reaction. So now we are using this new way to isolate the tissue-specific exosomes, and then to see whether be more precise with a higher resolution to look at how the different cell types that mediate this long-range crosstalk between different organ systems. So with that, I'm going to thank the people in the lab, and we have a very talented about 10 people in the lab, and especially in this Eric and Gangjian and Mingqian actually made the major contribution to this second project, and Xie made the main contribution to this UTOF1 stem cell oxidation study. So I want to thank my NIH and the AHE support, and thank you all for your attention, and I'll be happy to take your questions.